Hi, my name is Sonia Kovačková. I'm E&L expert for small volume parenterals at Nelson Labs Europe. Today, I'll talk about the necessity of E&L qualifications for the lyophilized drug products and the primary packaging. I'll also explain why there has been such a debate about E&L qualifications for this type of drug products, and based on our internal observations, I'll address some of the most common misconceptions and challenges that exist in the E&L field for the lyophilized applications. So first, we will take a look on what lyo containers are and some of the most common administration devices for and after reconstitutions. Then we will look into the EU and US guidelines and what do they tell us in the terms of what kind of depth is needed for E&L qualifications for primary containers. In the next part, I'll talk about the interaction mechanism between the lyo drug product and the packaging and the challenges and consequences it brings for the Leachables testing after reconstitutions. And finally, I will speak about the reactive leachables and the concern they might pose for the lyophilized drug product applications. Let's take a look on what we are talking about. The lyo containers and the most common administration devices for and after reconstitutions, we can divide in three different categories. So the first one is the primary packaging. In this case, a lyo vial system, which is composed of a glass vial and a rubber stopper. The rubber materials that are selected for this type of applications have usually low residual moisture content and low water vapor transmission in order to keep the drug product as dry as possible throughout the shelf life, and this is important for the stability of the drug product. The second category are the containers that hold reconstitution solution. And although the reconstitution solution doesn't contain any API, the E&L qualification will follow the same approach as for any other parenteral drug product, which is a liquid drug product. The third category are drug administration sets to administer the reconstituted solutions to the patient. This can be a disposable syringe or an IV bag, or a pump system with administration tubing set. Let's now take a look on what do US and EU guidelines tell us about lyophilized drug products. First, I will tell something about the situations in the US. I want to draw your attention to the Table 1, which was originally published in the FDA Guidance for Industry 1999 and later has been published in the revised version in USP 1664. This table ranks the packaging concerns based on the degree of concern associated with the route of administration and the likelihood of the packaging component and dosage form interactions. So what does this table and the modification tell us about the lyophilized drug products? As you can see, the degree of concern associated with the route of administration stays the highest. However, the likelihood of packaging interaction with the lyophilized drug product has been downgraded from medium to low. And this was based upon the observation that the interaction between the solid lyo cake and the material is limited. However, here we can see that the likelihood of packaging interactions with the dosage form has been downgraded from medium to low. The low likelihood of packaging and the drug product interactions for lyophilized drug product is based upon the observation that the interaction between the solid and the lyo cake and the material is limited, and that there is a limited direct contact between them. However, and we will see that later in the presentation, the mechanism of leaching happens not through solid-solid contact, but by means of outgassing. The EU regulations for E&L qualifications are covered in the EMA Guideline on Plastic Immediate Packaging Materials of 2005. This guideline provides a decision tree, and for solid active substances and solid dosage forms, the risk of interaction is low and generally does not require container interaction study. However, for solid dosage forms intended for inhalation or parenteral use, the lyophilized drug product may need additional study. So if you think of what I presented about the regulations and what is covered by the regulations in US and EU, you might want to think, is E&L qualification for lyophilized drug product necessary? And although it might seem that this is not really necessary and it might be optional, the current position of FDA and the European authorities is that they need or require in-depth E&L study on the lyophilized drug products in the primary packaging. In this part, we will take a look on the interaction mechanism between the lyo drug product and the primary packaging. Let's start with the most common misconception that exists in the E&L field on the lyophilized drug products, and that is there is no or limited interaction between the lyophilized drug product and the primary packaging. As you can see here on this picture, the lyophilized drug product is stored in a glass vial with a rubber stopper. The lyo cake is at the bottom of the vial and the rubber stopper is at the top of the vial. If you think that the interaction can happen only through direct contact, you might want to conclude that there is no interaction happening at all. But this is not the case. The rubber stopper contains a complex mixture of volatile and semi-volatile compounds which originate from the impurities or are formed during the production process. They get released from the stopper by means of evaporation and outgassing. Once they collide with the lyo cake, they will stick to it and accumulate on it because the lyo cake has high surface area and is also extremely dry. So on this slide, we will zoom in into the previous animation, but now a bit more in detail. So as I said, the Leachables compounds are being released from the rubber stopper. They travel through the inner space of the vial and then they collide with the very dry lyo cake where they will get absorbed and they will stick to it. Because of this accumulation, there is no equilibrium that is reached between the semi-volatiles of volatile compounds and the gas phase in the vial. That means that more and more compounds can be released from the stopper and further accumulate in the drug product. Additionally, there are some reactive compounds containing electrophilic reactive group which may further react with the API or with an excipient or with another impurity, and I will cover this part later in the presentation. And it's very important also to say that the extent of the outgassing will depend not only on the drug product and the storage condition, but also on the quality of the rubber or the grade of the rubber. Now we will look into what is happening during the reconstitution of the lyophilized drug product. By adding of the reconstitution solution to the lyophilized drug product, all the leachables that are present in the lyo cake are solubilized. These are mainly volatile and semi-volatile organic compounds which were earlier introduced through outgassing. Additionally, the reconstituted drug product can come in the contact with primary packaging, and through this shorter contact also non-volatile compounds, metals and ions may be released. And let's take a look what kind of consequences this has in the setup and the selection of techniques for extractables and leachable study. So if you only consider the long-time storage in the lyo vial, you will include volatile and semi-volatile compounds. But because of the reconstitution step, you need to include the non-volatile and metal compounds and anions, or the combination of those techniques. On this slide, you see the typical setup and the workflow for an extractable study. This was earlier in the day discussed by Denis Janke, and he focused on the individual steps of the extractable study. The extractable study on the lyophilized drug product or on the packaging for lyophilized drug product does not differ much from extractable study on any other liquid parenteral drug product. The same situation is for the Leachables study. What you might want to consider for lyophilized drug product is to add reconstitution solution to the setup of the study. On this slide, I want to show you a typical Leachables study setup for a drug product which has shelf life of 24 months. The lines in the blue represent the long-term conditions and the lines in the green represent the accelerated conditions, which is often needed as the time on 6 months is often submitted to accelerate the submission process. You can see on the slide that for every aging condition, there is a drug product in an inner container blank solution or blank reference added. And I will discuss this later in the presentation because this will be very important for lyophilized drug products. The last type of interaction we need to consider is what happens during the administration of the reconstituted drug products. So we use devices to administer reconstituted drug product to the patients. The contact is transient and short, and this means that no long-term accumulation will take place. However, the levels of leachables, organic compounds, metal and ions can be elevated after this interaction. So you might want to include the administration step simulation in your leachable study design. Let's take a look on the challenges and consequences of leachables profiling after reconstitutions. One of the biggest challenges of Leachables profiling after reconstitution is the unavailability of the true or genuine blank solution. The Leachables assessment is a comparative assessment between the drug product in packaging which was aged and a blank drug product which was aged but had no contact in the packaging. The peaks or the compounds which will be present in the sample chromatogram but will not be present in the blank drug product chromatogram will be labeled as leachables, which were introduced through the interaction of the packaging with the drug product. Having a good blank drug product gives us also a very good baseline reference for the study. But what defines a good blank solution? What it is and why is this the issue with the lyophilized drug product? A good blank solution is a Leachables-free drug product. A good blank solution is also from the same drug batch as the contact sample and is put on the control storage together with the contact sample. On this picture, you see a filling line for the liquid drug product. The blank solution is taken right before filling into vials. But what is happening for the lyophilized drug products? Why there is an issue? The liquid lyophilized drug product is basically a freeze-dried in the vial itself, so it will always come in the contact with the primary packaging. Here on this slide, I want to show you the relevance of having a blank solution. You might want to think, you know what, I will screen without the blank baseline. But what will happen in this case? On the chromatogram, you see about 75 compounds, the impurities which are reported and labeled as leachables. But the problem here is that you cannot really distinguish between the leachables and between the compounds which might originate from the drug product degradation or from the production process of the drug product. Let's take a look on how we can overcome the challenge of unavailability of the true blank solution. The first option would be to consider time point zero as a blank baseline throughout the complete study. This comes with some advantages. One of them is that using this approach, the stability and degradation of the drug product will not be accounted for. So this is definitely not an option which we would recommend. The second option would be to use upright position as a reference for inverted position. Because the mechanism of leaching happens through outgassing and not through the solid-solid interaction. For us, this is no option at all. The third option is to use lab-prepared or freshly prepared drug product that is containing all the ingredients. But again, it comes with disadvantages, and one of them is that the drug product degradation will be different in the solution when compared to the lyophilized drug product, which is solid. So we can use this option with some limitations. And the last option is emptying the lyo vial right after the manufacturing and store the lyo drug product throughout the shelf life together with the drug product in the primary packaging. However, we would have to find the right container for the long-term storage, and this might be a challenging task to do so. And it is also very challenging to keep the lyophilized drug product dry through the entire shelf life of the drug product. So as you can see, we have several options, and this is a non-limitative list, but the conclusion is there is no perfect solution. For every new request we obtain or we get from our clients, we need to look at it case by case. The lack of unavailability of true blank solutions brings some consequences and challenges to the Leachable study design. Because no true blank is available, we need to obtain a very good extractables data. In our Leachable study, we want to rely more on the target analysis of the leachables in the drug product with quantitative methods, or we can add a screening step, or we can also decide to report all the referential compounds from the blank. Every step has some disadvantages and advantages, and it is very important to justify the design for the individual drug products. There are three important aspects we need to evaluate, and that is to provide a trend over time, so to see an evolution in the concentrations of the leachable throughout the shelf life, to provide the correlations between extractables and leachables, and evaluate the possible reactivity of the reported leachables. Another observation that is mainly present in lyophilized drug product is that some of the leachables present in the lyophilized cake have potential to further react with the API or a degradation product of the API, or with an excipient or another impurity which is present in the drug product. So these compounds usually have electrophilic reaction groups, and they can react with nucleophilic groups of another compound or a therapeutic protein. So in lyophilized drug products stored in the glass vial with a rubber stopper, the most common reactive leachables would be the halogenated rubber oligomers. On this slide, we have an example of a reaction between the halogenated or brominated rubber oligomer with the degradation product of an API. So here a covalent bond is formed between the sulfonyl function and the rubber oligomer moiety. Not only the APA can react, but also other ingredients can further react with the reactive leachables. On this slide, we have an example of glycine. Glycine is present in the drug product as an excipient, as a bulking agent. Another example is a chemical reactions between the impurity of a solubilized agent, polysorbate 20, which is a source for fatty acids. We are not the first ones and only ones who observe this type of reactivity of certain leachables. I would like to draw your attention to the article of Amgen, of the R&D team of Amgen, and the observations they made for therapeutic proteins. Also, the R&D team of Nelson Labs did an extensive work on evaluating the reactivity of the leachables. If you want to learn more, please follow the presentations of my colleague Paolo Forte, where he will cover the results of this study and what kind of impact it might have for your therapeutic protein application. Let's look on the key learnings of this presentation. We have seen that the regulatory need of depth of performing an E&L study is ambiguous. The interaction mechanism between the lyophilized drug products and the primary packaging is not based upon direct contact, but it's based on the mechanism of outgassing. This may lead to the accumulation of leachables adsorbed onto lyo cake. In addition, adsorbed leachables may be reactive and may lead to the drug product degradation. So adding a screening step in your former leachable study can evaluate the occurrence of leachables reactivity. This can further assist in mitigating the risk of the occurrence of immunogenicity, especially if you have a therapeutic protein drug product. And although I mentioned this very briefly, it is very important to consider the right rubber quality or the right match of the rubber with your lyophilized drug product. You can always consult your supplier and ask for help. Thank you for the attention.